we'll start off with like the disease process or an infection. If you get a bacterial infection, kind of what the process is. And then when we're done with that, we'll get into the epidemiology terms, like what a pathogen is, what virulence is, that kind of stuff. Or get into it in more detail. So the normal microbiota, the term used to be normal flora, right? Somebody, I guess, didn't like the term flora. So now we use, right, technically it's now referred to as microbiota. They are bacteria found in most of the areas of our body, right? You don't expect to find them in the blood, the spinal fluid, right? There's a lot in our gut. I think there's over 1 trillion different, not different, about 1 trillion bacteria or so in our body. Now, a symbiotic relationship is what we would call the bacteria that are in the flora. They're not hurting us. We benefit from having the bacteria. So we call it a mutualistic type of symbiotic relationship. If you have a viral infection, right, it would be considered a parasitic type of symbiotic relationship. No viral infection helps us. But that's what we mean by symbiotic. This is a depiction of these are bacteria that are stuck on somebody's tongue. There's lots of them in our mouth. If you get rid of the normal flora in your mouth, what can happen? Thrush. Thrush is just Candida albicans, a yeast that's normally in there, but because there's plenty of normal flora, they don't overgrow. Now, normal flora aren't considered pathogens. They are opportunistic pathogens if an E. coli gets in your urinary tract, you don't want it there. So it's a pathogen there, but it's not a pathogen in your intestine. So you call it an opportunistic pathogen. A true pathogen just means whenever it's in our body, then it results in a disease. There's something's wrong. Some pathogens are more dangerous than others. So pathogenicity, right? A good example, COVID-19, right? It was very pathogenic and virulent. Nowadays, not so much. It's about as deadly or as dangerous to get an infection with that as it is with, say, the flu virus. So that's the degree that we use the term host. Most of the time it's human beings for us. Now when we start talking about, animals being zoonoses, right? They, like say, sheep can get anthrax, and then we can get the anthrax from the sheep. So we might refer to the sheep as being a host of anthrax. But for most part, right, we think in this section will be the host we refer to as being us. Now, most mammals are good hosts because we maintain constant temperature, we have many nutrients, and we maintain a constant pH. If you're, say a lizard or something like that, they're basically, their body temperature is whatever environment they're in. So they're less likely to get, they still have bacteria, but the bacteria they have would have to survive in different temperatures. These are places where you'd expect to find bacteria, and they're just about everywhere except, we're not in our blood. They shouldn't be in our spinal fluid. There is what's called the Human Microbiome Project, and they're looking for RNA or DNA in different body fluid. Now, what we'll do, the first part of this section is we'll go through what happens during an infection, and it's a bacterial infection for the most part. And then we'll talk about what makes bacteria pathogenic, right, what they use. If there's flesh eating, we'll talk about, you know, how they eat your flesh. So a bacteria has to get into your body. Normally it has to have the appropriate portal to do that. It attaches once it gets there, and then your immune system is going to want to, right, fight it at that point. So there's ways that the pathogens can evade the immune system. I talked about them a little bit before. And then at some point you say because of their presence, they cause the problem. Now sometimes it's not the bacteria that's causing the problem. The problem is that they are secreting enzymes or they're secreting toxins. And it's the enzymes and the toxins that are really the problem. So we'll get into those sort of things. And then there's an exit, you know, as well. So these are ways, portals are how pathogens can get into our body. These are, you know, just some typical skin, eyes, nose. And like I said, you know, you think if they don't get in the right way most of the time, then they're not going to cause a problem. Right? Our skin protects us from all sorts of pathogens. And the mucous membranes do too. We actually secrete antibodies in our mucus so that they can fight things like the flu, viruses, and bacteria as well. So the term infectious dose is how many pathogens, it can be number of virus or the number of bacteria that must get past or into the portal. And if you've got less than that, then right, it's not enough. So for instance, the gonorrhea, they did some studies and found that it took about 1,000, give or take a few, I'm sure. If you use the term ID50, it stands for infectious dose, and that tells you, right, you have a 50% chance if the ID50 is 20. If there's 20 bacteria or virus that get into your body, then you have a 50% chance. And that's probably a better way of describing it. Your chances, the attachment, right, they get in, then enough of them get in, then they need to attach to things using fimbriae, and even the capsule can help them, especially in the lung. Right? I think I told you before, right, once the bacteria gets into the lung, then if they don't have, you know, a capsule, then your immune system are, right, you're going to expel them. Well, the microbes have a ligand, right? That would mean, you know, like if there wasn't a capsule. Now, a ligand is kind of like, we're going to talk about antigens in section 13. You can think of a ligand as an antigen. Ligand just means it's the thing that sticks to some type of receptor on your body. So you think of the body as, or the host, as a receptor that something on the pathogen can stick to. Then once the bacteria gets in, it's going to colonize, right, which just means grow in number, and then establish an infection. And your body's going to do its best to keep the bacteria right where they're at. That's why you say, like in the skin, you might get a boil, right? It kind of cordons off the bacteria. Then you have, because they invade your tissue, that causes problems. So the evading phagocytosis, so we talked about this before, the capsule, right, can actually cover up you know, like an antigen on the surface. So that a white blood cell comes along and it's looking for a foreign antigen, and the capsule protects it, sort of like hides it from them, right? So it doesn't appear to be a problem. And also, if this white blood cell was a phagocyte, the capsule would prevent it from attaching to the bacteria too. And then there are actually things in cell wall, they call it, you know. So that's not the capsule, but the wall around the bacteria can contain things that prevent digesting. Means when it gets into the white blood cell can't break it down. Right? And that's the problem when you have a Mycobacterium tuberculosis infection. It does it. You can't use phagocytosis to kill it. You can have it for a long time. So what's antigenic variation? I like to just bring this up because it's important, and we'll talk more about it in the following sections, right? But that's what happened to COVID-19. Right, it went from Omicron, it came Delta, Beta, Alpha. Those were antigenic variations of the COVID-19. So it goes from the shape changes through mutation. An antibody that sticks to the square antigen, if it changes its shape to this other shape, then this antibody no longer is effective. Right? So that's what we mean. Mutations cause proteins change. Right? That's the variation. Antigens that cause antibody production. That's, you know, that's what an antigen does. But if it changes shape, then the antibody may no longer be effective. Just like even the original COVID-19 vaccine. Like there was talk a year ago, well, we got to change it so that the antigen that it makes in your body represents the newer versions of COVID-19, and then somebody decided so microorganisms, these, and we can put COVID-19 on here, it's basically RNA viruses. The way they replicate, if they're RNA, that they're more likely to, like HIV is RNA. This is a protozoan that causes problems after sleeping sickness. So as I said before, I tried to write this on the board, this concept of phagocytosis. And in section 13, we'll talk about neutrophils and macrophages are white blood cells that phagocytize bacteria. So if bacteria secrete enzymes, right, we talked about, you know, if they secrete amylase, they can break down starch, but they can also secrete enzymes, right? That means that the enzyme gets out of the bacteria, and then that enzyme can cause problems. Right? If there's glue holding these two epithelial cells, basically it's usually proteins. If the enzyme breaks those proteins down, then the bacteria can get through the epithelial layer, right? And the epithelial layer, its purpose is to protect you from pathogens, right? It lines all your mucous membranes. So these are some other. If you say a bacteria is flesh-eating, they don't have little mouths that are eating your flesh, right? They're releasing enzymes that break down things like collagen. Right, that's a protein in your skin. It holds cells together, right? And so you break down that protein, then you destroy the flesh. You know, if you don't cut out the flesh and the bacteria, it just keeps spreading very quickly. You know, they're secreting enzymes that can move faster than the bacteria can. Hyaluronidase is another enzyme that breaks down hyaluronic acid. Then you have this guy dissolves clots, coagulase makes clots. So they just disrupt the clotting mechanism. But this is used, right? Streptokinase is helpful for people that have strokes. Treat it quick enough, you can dissolve a clot before it causes irreversible brain damage. But that's eventually what probably would happen. So enzymes can be secreted. The other main problem, some bacteria secrete toxins, right? Which is pretty self-explanatory. They're toxic. There's two types. There's an exotoxin that's secreted, and then the other one, there's one endotoxin that we talked about a little bit in section 4. It's a component of gram-negative cell walls. Lipid A is the other name for it. It doesn't cause big problems, but it can make, you can get a fever from it kind of thing. It's not as dangerous. The exotoxins are the ones that are more well known. We talked about them a little bit when we did the lysogens. So they're protein, they're heat labile. Because they're proteins, they're very potent. You don't need much. If you're given Botox, it better be diluted correctly. It could kill you. And they have a specific target tissue. So I'm going to get into the 3 categories. I'm going to mainly just talk about the first one. The other 2 I'm going to briefly talk about. But we mentioned the botulinum and the tetanus. Clostridium tetani is the bacteria that produces this toxin. Both the botulinum and tetanus are neurotoxins. So that means they only affect neurons. And they only do that because they bind to a receptor that's only found on neurons. Now the cholera, the toxin is an enterotoxin, meaning that it affects epithelial cells in your intestine. So when it gets into those cells, then these cells in your intestine secrete water into your intestine, right? And it's supposed to do the opposite, right? Your large intestine is there to absorb as much water as it can, from your feces. The tetanus actually causes neurons to fire and contract muscles. And all the muscles in your body contract. Awful. And then botulinum does the opposite. It blocks. That's why, so your muscles, they paralyze the muscles in your forehead. They're called AB toxins because there's 2 subunits in the protein or the toxin. One binds to the receptor, the other part of the protein elicits whatever response they have, right? It's how the toxin works. So category 2 are, they're referred as membrane disrupting because they cause cells that they affect to lyse. So a leukocidin affects white blood cells, and hemolysin affects red blood cells. In lab, we use the blood agar plate that contains red blood cells, and then Streptococcus is a bacteria that causes strep throat, so it's useful in identifying, right? But that's why you have alpha and beta hemolysis on the BAP plate, is because the bacteria secrete hemolysins. It's actually a toxin. Category 3 exotoxins provoke an intense immune response. And a good example of this is when you have the COVID-19, right? There's parts of that protein, right? It's been engineered in a lab. There's 3 parts of it that make it more pathogenic because it provokes an intense immune response. And people die from that, not so much from the virus, right? It's the response to the virus that kills you. That's the kind of the problem with if you have allergies to peanuts, it's sort of like an intense immune response as well. It can kill you, right? You get, anaphylactic shock. But they call it a cytokine storm is another word for this, where there's so many cytokines being released from white blood cells that your body just doesn't know what to do. Toxoid is when, like if you get the vaccine for tetanus or the vaccine for diphtheria, the vaccine produces antibodies to the toxin of these bacteria, right? It doesn't protect you from the bacteria, right? Because the bacteria aren't hurting you much. It's the toxin that they secrete. So an inactivated toxin means that they've treated it so that it doesn't produce the effect that the toxin, the poisonous effect of the toxin, but it still elicits your immune system to make an antibody. So that if you get an infection and the bacteria secrete the tetanus toxin, then those antibodies neutralize the toxin. Right? If you bind antibodies to this bacteria, you could say that you're sort of neutralizing that as well. Then the endotoxin, right? It's only found in gram-negative bacteria because it's a component of their cell wall. Cytoplasmic gram-positive, right? LPS is lipopolysaccharide. That's a component of the cell wall of gram-negative. It only is a problem normally when the bacteria die. So if they're dying from antibiotics, let's say you go to the hospital and they're trying to kill the bacteria with IV antibiotics, probably within an hour or two after getting the IV, this endotoxin is released by the dying bacteria, and your blood pressure goes down, and you get spike of fever that you didn't have prior. It's all caused by the endotoxin, right? They wouldn't be too worried. They kind of expect it. So portals of exit, pretty obvious, nose, mouth, just everywhere, and then just like in portals of entry, but you don't see feces, urine, or semen. Vaginal fluid, you could add that to the list, I suppose. Anything that's secreted, spit. Right, half of this section is basically the course of a typical bacteria infection, what happens during that process. And the rest of this is going to be on epidemiology or epidemiological terms, like things like a systemic infection refers to, right, throughout your body. A local, it may start locally, I mean that's usually what happens, and then it gets into your bloodstream, gets into places it's not supposed to go. So you kind of want to treat it quickly. A primary versus secondary infection. Primary means is the one you get first. That's of course, hopefully you understand that. But if another bacteria or virus that didn't cause the original starts after the first, then you call that a secondary infection. So, a good idea for that is like say you get COVID-19, and many people may have gotten a secondary bacterial infection along with it. You know, so it's not always factors that influence spread. We'll go over what a reservoir is. That's basically where you find the virus. If there's, mammals in Florida that have rabies right now, then they are considered a reservoir of the rabies virus. But portals of exit and entry, and then we already talked about them. And then there's modes of transmission, and I'll cover those. So the definition, or a simple definition, reservoirs of an infection is the source of the infectious agent. If you went and ate food and got food poisoning, then that's the reservoir. Usually, like you think of a reservoir, it's something that sticks around for some time. Now, there's a difference between, we are animals, but in this case, humans reservoirs, then we're thinking of human being as the host in that sense. So animals are different. But the reason, HIV is spread or hepatitis C is spread is because there are people that are carrying it, right? So they act as the reservoir in that case. That's pretty self-explanatory. Then there are animal reservoirs, and sometimes, like with rabies, the virus that causes rabies causes the same disease in the animal that it causes in human beings. But if you have, say, like the tick that bites you, it carries Rickettsia rickettsii, Lyme, but it's a bacteria. The tick doesn't come down with Lyme disease. Right? Lyme disease is really complicated. It's different people, but it's a carrier. So when you think of zoonoses, those are usually animals that carry the disease, and they get the disease as well. So even anthrax, sheep can get the same kind of disease that anthrax causes in us. Will cause the same symptoms in the sheep. Then there are 3 types of mode of transmissions: vehicle, vector, and contact. Then there are 3 types of contact transmission: indirect and direct. And then this is droplet, which is at less than a meter. But that means when you cough or sneeze, that the droplet can go that far. Indirect would mean you coughed and it landed on somebody's desk, and then you got it through the desktop. That would be an indirect. And then direct would be person-to-person touching or sexual contact, something like that. You can get it parent to child, and that's vertical. That means, mother has HIV, the baby has HIV, that kind of thing. That's vertical. Fomites are objects that spread the pathogen. But that would be considered an indirect type of contact transmission. Then we have, now just reiterate what vehicles are, right? When they say waterborne, right, it's spread through the water. Airborne, spread through the air, right? You're describing a disease. Foodborne would be a good one for that. Are all sorts of the food poisoning. Usually it's insects when you think of vector, but I think that it could be other things too. But normally you think of insect bite as a vector, and you call the vector if it's you know, a mosquito. It might be a specific genus and species of mosquito. If it's mechanical, then you're not being bit by the fly, it's landing on you. And it has maybe some fecal contamination on the legs. And then it lands on, say, you know, potato salad. It's not going to leave that many bacteria on the potato salad that'll cause a problem. But if that potato salad were to say, you know, let the bacteria grow for 3 or 4 or 5 hours, or like say you don't keep it refrigerated for any length of time, then enough bacteria grow so that you get enough infectious dose. Mosquito, these are some examples of, you don't have to worry about this table, right, but it's only the Anopheles, that genus of mosquito transmits malaria. Other types of mosquitoes don't. So when you have like the Chagas disease, or African sleeping sickness is another one. There's a fly, and it's that specific genus of fly that transmits it. Other flies don't. So it's weird how that works. So that means that, you know, not every mosquito bite is going to spread dengue or yellow fever. And it usually means that when the pathogen gets in the mosquito, it's able to multiply in the mosquito, so that's problematic. You're not going to get, so if someone gets bitten by, has HIV, gets bitten by a mosquito, there's not going to be enough HIV in that mosquito when it bites, so if it bites somebody else, to spread it to somebody else. Yeah, epidemic, everybody's pretty, we've heard of that. Now, the pandemic was kind of odd with COVID-19 too, that they waited about 2 or 3 weeks before they, it was a pandemic, but they wouldn't say it. They wouldn't use the word. I don't know why they waited so long to do that. But that means that it's gone from one continent to another, right? So then it's considered worldwide at that point. If we have the flu outbreak in the United States, it may start in the cities, but it's hard to define what like why is it different? You say it's endemic in this instance, like if we constantly see a certain bacteria in this region of the United States, then we say that it's endemic in that part, or maybe a state. Normally it's not a state or city. Rabies is endemic in the United States, but it also might be endemic in France, but we don't consider it a pandemic. And then just to give you an example, rabies is endemic in the US. It's not the only place it's found, but that's how you would describe it if you were, if you looked in a textbook and they were talking about rabies, that's what there would probably be a statement about that. And a common source is good example of that is food poisoning. It's not going to be spread from person to person when that happens. So you might get 100 people that get sick. Within a couple of days, though, there's none, right? They get over it pretty quickly. Propagated is like the flu or COVID-19, right? It gradually spreads up, or it can go up pretty quick, I suppose, too. But you get a long duration, it can be months. Morbidity rate. Morbidity is the number of diseases. The mortality is the number of deaths per 100,000. So this gives you, it allows you to kind of compare, is it more common in the United States versus, or like say one state versus another state, gives you an idea of the percent of people being affected. It's kind of another way to think. Versus secondary infection. Opportunistic pathogen means that it's a bacteria that's normally normal flora in your body, and it becomes a pathogen because it gets in the wrong spot. Or C. diff is a good example of that. right? You have an infection, they treat you with antibiotics, and it gets rid of so many bacteria in your intestine that the C. diff that's normally there as normal flora becomes a pathogen. So the septicemia means bacteria dividing in the bloodstream. Bacteremia, there's bacteria in the blood, but it doesn't refer to them being dividing. I'm not so sure that that's ever a case, but that's, bacteremia just means they're present. And toxemia, there's toxins in the blood. Viremia, viruses in the blood. Those are just terms that are used. So these are 4 methods to control the spread of communicable diseases. Right, we got familiar with patient isolation or quarantine. And you know, hospitals will do that normally and expect you maybe to wear gowns and gloves into a certain patient's room. Now, of course, immunizations. You know, unfortunately, you know, the vaccine for COVID-19 really hasn't done a whole lot. We've just been lucky that it's become less and less pathogenic. And then if it's spread by a mosquito, you know, you can maybe spray. I don't think we spray enough around, you know. Healthcare associated. That's the new term. And that's the term you might see on the test instead of nosocomial. I think I've changed everything now. The nosocomial or healthcare associated, this helps you actually understand the concept better. You get it at some medical facility. Normally you're at the medical facility because you're sick, so you're more likely to contract an infection there, you know, because you're somewhat compromised. You have more microorganisms. So to control them, right, it's always important for a hospital to do what they can to, right, have some method of keeping this to a minimum. But control of aseptic technique, of course, education of staff. I guess like this would be a good example of it. Washing your hands, gloves, pretty straightforward. And then this is an old table. I forget the year this was. I think this is about 10 years old at this point.